In treatment, I think one thing would be is most of the therapy is now we are focusing on the metastatic setting. For example, immunotherapy and FGFR or Enfortumab. So now if we can bring it to the neoadjuvant or adjuvant setting, and will that have an impact early in the disease process? And that would be something very critical to understand. We had a clinical trial where we showed benefits of combinatorial anti-CTLA-4, anti-PD-1 approach in a neoadjuvant setting, and others have also conducted trials demonstrating the similar efficacy of neoadjuvant immune checkpoint therapy. Now, another setting would be if there is a patient who got neoadjuvant chemotherapy for localized bladder cancer and had cystectomy, but on cystectomy we do see a high risk pathology, then if we find any certain genomic alteration like FGFR-3, then should we treat them in an adjuvant setting? So number one, I feel understanding the treatment in different settings of bladder cancer in the neoadjuvant, adjuvant, and metastatic setting of this available agents would be critical. So second thing, as I said, is the timing, right? Because as we know that every drug that we give, it changes the microenvironment, the tumor microenvironment. So how we can better sequence them so that when we go for the one therapy to the next therapy, is it going to positively or negatively impact the next therapy? So that would be important to know, like how to sequence them. Third, one major question would be like, as I said, like understanding like the predictive biomarkers for these different therapies, and so that we can do a better job in patient selection. And that would also help us with the toxicities, because these new drugs also have distinct toxicity profile, so that we understand this toxicity profiles better to kind of improve patient outcome. And I think those are the things, and definitely like we need to have ongoing research to find what are the other new targets that would be beneficial in the treatment of bladder cancer.